Hi everyone, my name is Jason Fung and welcome to ZEISS Microminutes. We're here live at ZMCC in Dublin, California, and today I'll be talking to you about neuron segmentation using AI with our ZEISS arivis suite. And so again, I want to extend a warm welcome to our ZEISS Microminutes. And here in the link, the QR link, there's a list of upcoming ZMCC workshops that may pertain to your research and academic technologies. So I'm going to give you a brief overview of what our ZEISS arivis solutions has in store for you. And so arivis is a flexible software, meaning that it is able to take in over 50 different imaging file formats and lets you analyze any images from the scale of organisms all the way to puncta in any different modalities, so such as wide field all the way to lattice light sheet dealing with big image data. And so arivis is AI powered. So what does that mean is that imagine that you could use pre-trained publicly available deep learning models, for example 𝗁 that could segment your cells individually and train your own state-of-the-art custom deep learning model such that that's tailored to your imaging datasets in the cloud. The beautiful thing about our software is that it's multidimensional, meaning you can look at 2D images all the way to 4D from the scale of if your images are from megabytes and all the way to terabytes of data. So if you're working on large 3D time-lapse images, arivis could be the solution for you. And just to touch on one last bit, but for those who are in the type of environment that requires high throughput analyses to be done, we have the arivis hub, which is our server-based scalable processing solution, which allows you to parallelize your image processing and analytics such that you can get from multiple images to results way faster. So all in all, arivis is a very future-proof software such that it's flexible, handles multidimensional data easily, and we have scalable solutions for you if you need parallel processing. And so we understand that biological structures are hard to segment and analyze, and finding the correct method for segmenting your cells is always a huge hassle. So deep learning is now at your fingertips. We have arivis Cloud, which previously used to be called 𝗶, which allows you to train your own deep learning models by directly interacting with your own data set. And training is just as easy as painting some cells or structures of interest and just clicking train. And so this is all fully done on the cloud, and your model is now yours to own. And before you know it, you can reliably use that same AI model over and over again without having to painfully find the correct processing parameters to segment your complex biological structures. And as imaging conditions change, you can reuse that same model and apply it to your new image data sets such that it can learn to analyze your images from image to image variability. And so you're utilizing our cloud infrastructure that allows you to do fast model development. And this whole entire process is no code at all. So it's for the everyday scientist such that you can just immediately jump in and use. These deep learning models can also be downloaded to segment your images offline in Zen or even in our arivis software packages. So just to conclude, our ZEISS arivis solutions in a nutshell is that you can perform routine analysis tasks on simple 2D images all the way through big 4D datasets, and complex tasks such as tracking and AI-assisted segmentation is there for you to use. Our software is also Python extensible, so if you have already pre-written Python scripts that you want to integrate into our software and utilize our visualization material, you can do that as well. And so because our topic today is neuron segmentation and reconstruction using AI, we have neuron tracing, which allows you to perform statistical analysis to get your morphological data for downstream analyses. Okay, so getting to the meat of the topic today, I'm going to be showing you a complete end-to-end ZEISS solution for neuron reconstruction with AI using arivis. So a couple of webinars ago, our application scientist, Amita, has showed us the power of the Axioscan, providing amazing resolution of whole slide images, such that we were fortunate to have the opportunity to image whole slides of Golgi-stained neurons of mice brain. So let's take a step further. What can we do with these images? What if you dig deeper into your whole slide, you see these neurons and you want to quantify them? How about tracing them? How about analyzing the structure of these neurons? How would you even go about it? And so neuronal morphology is often difficult to analyze due to the complexity in identifying and segmenting features automatically so that you can perform your downstream analyses. And for us, it's really easy to distinguish by eye, such that the really small bits are spines, the long parts are neurites, and the big blob there is the neuronal body. But the computer doesn't know that. Traditional methods, they don't know what these are. So some of the challenges is that there's no clear intensity distribution that's creating the boundary of these features. The focal planes are also can be out of focus as well, and your image conditions could be changing from sample to sample. And so all of these coming together requires a lot of heavy pre-processing and a lot of expertise to even extract these in a using classical traditional methods. And so how can we quantify our images? So the approach would be to segment the different components of these neurons. But then you would ask yourself, isn't detecting these features automatically and accurately a really hard problem then? Well, ZEISS arivis solution has powerful AI platforms on the cloud, such that an everyday scientist can train their own state-of-the-art custom deep learning model that's tailored to solve specific challenges. For example, segmenting the neurites on the left, where I'm just painting a couple of neurites, not even the entire branch, and on the right, I'm just labeling some spines. So just like a coloring book, just go in and you just draw. And all I'm doing is telling our model what is or isn't the structure of interest, and training is as easy as just doing this. Painting and clicking train as just like easy as clicking it as a button. And so here I'm showing you that even further, you don't need to annotate everything to train a state-of-the-art model. All I did was annotate a couple of neurites in a couple images, and after you train it, you can preview your segmentation. So this allows for quick and accurate model development for your applications. So with these trained models, it's easy to just import into arivis to perform your segmentation, rendering, and visualization of your segmented objects in 3D. And the important thing is that you can extract these data, such as the spine volume or distance-to-distance statistics between these spines labeled in red. So for example, if you're looking at what's the mean volume of these spines, you can get these statistics really quickly, and also even doing distance to distance as well. Or even how many spines are there in this region of interest. Okay, so what about cell-to-cell statistics? Using your AI model, we can pre-process our image using our deep learning model and the objects that were created, such that we can focus we can process these images to focus on features that really matter, such as the cell body, neurites, and spines with our reconstruction. And here I'm just showing you the landscape of the segmentation in 2D. And so after performing our processing of our bright field images, so it's not just in 2D, it's 3D as well. And so we can use our AI segmented objects to focus on the important features so we can trace our neurons. And so here in our new 4.2 release of arivis, you can preview your image, you can preview your traces in 3D, and you can check it along with the reconstructed image from our AI reconstruction. This is to ensure that we can get accurate tracing and make sure that the tracing is what we expect to see. And so these segmented objects and traces are interactable, allowing you to click on your traces, check out the landscape. Here I'm clicking on an object and opening up the data table. In this way you can look at single cell statistics. And if you want to look at a summary of it, you can also look at you can basically look at from single cell, individual objects, all the way to a whole population for further investigations and analytics. As you can see, these are the common morphological data that you would get, such as the length of the neuronal structure, your neurites, or even your tortuosity. So all in all, using ZEISS's arivis Cloud AI platform and advanced image analysis software, arivis Pro, you can analyze, interact with, and render your results for publication-ready materials quickly and easily. And as this video shows, as video shows, you can segment your soma, your neurites, and transform all your objects into neuronal traces as your reconstruction. Okay. So that was a quick live demo of our arivis software for neuronal reconstruction and segmentation. And so here are some useful links and resources that you can scan to look at what our upcoming ZMCC workshops are, scan for our ZEISS portal, and upcoming live streams to look forward to. And thanks for tuning in. Feel free to ask any questions, and I'm here to help you answer them. Okay. Thank you everyone for watching, and see you in the next live stream.